today. Yeah, it's coming up, great. And sorry about this Merck KGaA Darmstadt, Germany thing, but I'm obligated to do it this way so you don't confuse our company with the American Merck. So yeah, in the US you always have to give the talk like this. But now we know that, and yeah, I can just happily move on and share our experiences of using free energy calculations in our in-house drug discovery projects with you. And I think we have collected some interesting data, and I'm happy to share them today. So first of all, I would like to thank everyone involved in this. First of all, of course, our medicinal chemists in Darmstadt and Boston, who in the end synthesized and tested all these molecules, the computational chemistry and biology group in Darmstadt, and our colleagues at EMD Serono, who worked on the modeling side, and our colleagues at Schrödinger with all their support for using FEP+ and also for partially running some of the calculations. And we've also been looking a little bit into absolute free energy calculations in collaboration with Levi and John, but I won't be talking about this today. So today I will take you on the journey of our 2 years experience of relative free energy calculations with Schrödinger FEP+ in our in-house discovery projects. And well, it has been a little bit of an up and down, and I'm happy to share some of those experiences. We have looked at a broad variety of targets and series. We've looked at 25 targets or projects and discussed whether or not we can use FEP. Out of those, we have been initially able to validate 10 targets. We've run over 25,000 FEP perturbation calculations and obtained from this 5,000 final predictions. That doesn't really, that's a lower number than the number of compounds for which we have any kind of predictions, but that's kind of like the higher confidence predictions. And out of those, more than 350 compounds have been synthesized and tested, so we can compare the predictions against the experimental data. And this gives us a good data set to evaluate how does FEP+ perform in our in-house drug discovery projects used prospectively, so from a blind prediction. What can we learn from this? What are the limitations? And what is also the impact on the projects? So first of all, let's have a look why we didn't use FEP in so many projects. And we tried to see what was actually the reason for that. And indeed, the major reason for this is the availability of a co-crystal X-ray structure. So this is lacking in those projects for various reasons, and that's why we cannot use FEP there. Here's also an overview of all the validation studies we performed. So from several targets, as you can see, we have validated or have tried to validate multiple series or sub-series. You can see that this distribution is rather broad. On the one hand side, we are able to get a lot of series for which we can get really nice validation results below 1 kcal per mole. But there's also a big chunk of chemical series for which the validation result is rather so-so in between 1 and 1.5 kcal per mole. And obviously there are also some series where it doesn't work at all. Interestingly, also for some targets, like the yellow one here, we have series where it works nicely and series where it doesn't work nicely. So it doesn't only depend on the protein target, but it's also very dependent on the chemical series. So one of what's also interesting to look at is why is there any factor that can tell us if a validation study will work or not? And typically one of the things people think about that's really crucial for making FEP work is having really a high resolution crystal structure. But when I look at our data, at least, it doesn't seem to correlate at all with how good the structure itself is. It's more of an intrinsic property of the system. When we are using FEP in real-life drug discovery projects, the systems are never perfect, and we are always facing uncertainties in protein and ligand structures. I try to list this up as a more qualitative judgment of what problems are we actually facing, and we can really see that in almost every project we'll have something that could potentially limit our accuracy in free energy calculations. We have projects where we can see that there's protein flexibility going on in reaction to the ligand binding, or we suspect at least that that is something that's going on. We have, like almost every project that we tried, we have missing parts, larger missing parts in the X-ray structure that have to be modeled, and of course in this way bring an uncertainty into the system. It's also common that throughout the course of the project, when we move in chemical space, that we get more and more uncertain about the binding mode of the ligand when we add new chemical groups. And another thing that often happens is that at some point in the project we also will be a bit uncertain about the protonation state of the ligands, or at least for a subset of those ligands. And this of course also affects the quality of the calculations. We have also seen that there's still room for improvement, and I'm picking out one example where we can nicely see that there is room for improvements in the force field. So the first calculation I'm showing here is a validation study run with the standard OPLS-free force field and the standard atom typing. And as we can see here, the results don't look that nice. And especially when we looked into the data, we saw that when we changed this bicyclic core of the molecule, we cannot capture these changes well at all. And that made us that brought us to think that probably the partial charges on the bicyclic core are maybe not well represented in this standard approach. And since our colleagues pointed out that there is a new, at that point still better version for calculating the partial charges from quantum mechanical calculations and then using those in the simulation, we decided at that point to try that out. And indeed, in this example, using exactly the same ligands, exactly the same binding poses, we got much improved results with these new partial charges. Yeah, and I think this is coming out soon in one of the releases, and this is for me one of the few examples where we could really pinpoint it down to a force field problem. We also looked at other force fields briefly. My colleague Tim used the Silks Bio CGNFF program and ran it on our entire in-house database to see how well those CGNFF parameters would potentially cover our in-house chemical space. He found that for 45% of the molecules, the program gave back a parameter penalty of below 100, which is estimated to be reasonable, so the parameters should match quite well for those molecules. On the other hand, that of course means that for the majority of those compounds, one should consider at least looking at the parameters or maybe also reparameterization. We've also seen this in using OPLS-free, that almost in every project we have run so far that we had to do extensive quantum mechanical calculations to reparameterize the torsions. And these things together for me suggest that maybe the force fields have been built on a chemical space that doesn't totally overlap with the in-house chemical space we are seeing. So it would be interesting to think about how to go forward with force field development, how to maybe somehow get access to this different chemical space and also parameterize the molecules in a better way. So now on our own FEP plus results. So here I'm showing the results of our validation studies in the 10 projects and the results of the prospective data we've acquired so far. So indeed we got nice results for those 10 projects in the validation study with only very few outliers outside of 2 kcal per mole. When we look at the prospective data, you're probably looking at it and thinking, so, yeah, well, it looks a bit so-so. It's definitely higher. The MSE is definitely higher than what would we have liked to see. Although when we put this into perspective with a more standard approach, like using a docking score for those 5 of those projects, I just did this as an exercise. Or also for putting this into comparison to MMG-BSA, we can still see that there's a lot more signal in the FEP calculations than in those 2 simpler methods. Although one has to say, at least for MMG-BSA, for this red project, there is some ranking performance, but overall it's like a really nice point cloud. So these are the results of those 10 projects, but we have also gone a little bit through a learning curve throughout those 2 years concerning those different projects. In the beginning, we were very keen to apply FEP to projects, and we were like heavily looking for any project we could throw it at just to try it out. So we were also accepting projects in the validation study that had an MSE above 1 kcal per mole, between 1 and 1.5, and maybe already showed some limitations in terms of there's one pocket where we don't get good accuracy and stuff like this. However, when we looked at this back, we said, okay, we need to be stricter in the future. And so this is the data for the 5 most recent projects, where we said we will only accept those series if we got really excellent results in the validation study. And here we can also see that the first prospective data we have obtained for this is looking better than before. Generally, what we have seen when using FEP prospectively in the projects is that the prospective accuracy tends to be lower than the accuracy we see in the validation study. This is, of course, expected also, since we are, first of all, we have more uncertainty when we apply FEP prospectively, because we don't know everything about the system. We haven't studied it well at every time point, and we don't have maybe all the information available at a certain time point in the project when we make the prediction. We are also most interested in predictions that are like the most extreme predictions from our data sets. So either finding the very best compounds or finding the worst. But rather, most of the time we're not interested in those molecules that are most predicted to be very similar to the reference, but rather than the ones that are different. And obviously for those extreme predictions, the chance of uncertainty and error, less accuracy is also higher. And generally, we also, when we do calculations, we are often faced with a situation that we have an idea for a new compound, and then we already know that that's probably already pushing the FEP technology to the edge, because it's maybe a quite large transformation. But I mean, in many cases, you still want to at least try it anyway. So when you use FEP prospectively in projects, you're almost always pushing the technology to the edge. I just want to share some of the observed limitations when we were running it on all these different compounds. Unfortunately, I can't really make these observations very quantitative, so this will just be a little qualitative storytelling. So first of all, we have often seen in validation studies that predictions are quite good for one modifications in one pocket, but might not be good for the other part of the molecule. We've also seen that it's difficult, in many projects we've seen that it's difficult to predict, or more difficult to predict the parts of the molecules that are more solvent exposed, so at the open side of the binding site, or even sticking out into the solvent. We've seen that especially when you didn't have anything in your molecule before and then you grow something out of there, that's often overestimated. It's predicted to be a lot more potent in our experience than it usually is. It's also dangerous to have such a prediction in your map, because it pushes all the other compounds down. For me, this indicates that there might be also some more work to look at the water models, because water is probably too easily displaced. Obviously, I mean, for many people this will not come as a surprise, but also doing this very common transformation from a very short substitution to this long flexible change, we've also seen that this still gives sampling problems, and I think there's potential to improve that a lot. The same, in a similar direction, is that we have seen that we got errors when we went from this rigid aromatic system to the aliphatic rings. It was easier to even remove the aromatic ring and get still reasonable accuracy than to go back to this very flexible ring system. So my suspicion there is also that it's not sampled properly. And last but not least, even though in FEP Plus there's now the possibility to do, and an advanced scheme to tackle free energy calculations with net charge changes, or with changes in the moving the charge around on the molecule a little bit, like shown here in this example, we've still seen that this gives larger errors, and even if the trend is correct, the effect is almost always overestimated. So there's also some work to do here still. So now Mark has already given a really nice introduction into this topic of the more operational side, and I would also like to share with you some of our operational learnings. So when we started off with FEP 2 years ago, the idea of using it was more like this. Yeah, we will continuously evaluate all the synthesis proposals and then prioritize them with FEP. So for example, in 1 week we will have 20 to 30 compounds in that project, and then we will make the best 5 of this or something like this. So here's an illustration of this from one of the projects. So here we evaluated 32 compounds of FEP. Out of those, 22 were synthesized and tested. And one could have avoided to make at least 10 of those molecules. The benefit obviously would be to save synthesis resources spent on those inactive molecules and focus on the promising molecules. And however charming that concept sounds, we have unfortunately experienced that this causes us a number of problems when we really want to use it in that way. So first of all, often when you come up with this, yeah, please every week give me your compounds, the number of compounds that will be really proposed by the people is unfortunately not always that much larger than the compounds that will be synthesized, which obviously limits the impact of a technology like FEP. Then there's also this psychological effect. Once you have put down on a slide, on a Word document, on a database, all your new nice ideas, a lot of chemists think, oh, that's a really nice idea. Oh, I will just make it now. It's I have some capacity in my lab. I don't want to wait another week or so to get those predictions back. And that's, of course, obviously also not good for the impact of FEP. And then there's a long-term psychological caveat. That's after 6 months, nobody will remember the bad molecules that were not made. But everyone will always remember the one good molecule that solved the problem in the project. So taken all together, the scheme of prioritizing synthesis proposals might sound very attractive, and it's easy to incorporate into the standard MedChem optimization workflow in a project, but it's actually having only a very limited project impact. So we thought, what can we do better to tackle those problems? And first of all, we want to be more surprising and use FEP really to find novel active molecules. And maybe also chemistry that wasn't really directly on the radar of the project team. Secondly, we need to be quick. And quick is, 1 week is not bad, but it's actually still too long. And the only way to get around that, because you need some time to prepare those calculations, set them up, and analyze them, is to really go into front-loading a large number of calculations, so you already have them ready for the project team. And this all culminates together into going big and performing large-scale FEP calculations on custom-built libraries for the project, according, of course, whenever this is useful in the project and when it's also possible from the science point of view. So here's an example of where we first tried this out in a project. So here in this project, the team was looking for a replacement of a 1R group and wanted to replace it with a ring system. And out of curiosity, we thought like, oh, can we be very systematic and cover everything that exists? So we said, okay, let's go into all the databases and collect all the rings that are there in the world, filter them down, and then we came up with this library of 1,000 compounds. And we ran FEP calculations on them. Indeed, many suitable replacements were suggested by these calculations, which at that point in the project came out as a surprise, because before we had trouble replacing this part of the molecule. Some of them were more conventional, and some of them were, let's say, more rare ring systems that didn't always meet the enthusiasm of the chemistry team. And out of these, a few selected several examples have been synthesized. And more overall, the agreement between experiment and prediction is quite good. So what we learned from this is that, first of all, we were able to actually deal with this large number of calculations also in-house. And actually just doing this, for me, this was also a rewarding experience. So I learned better how to use this tool, how to more efficiently process those data. So this was a good thing in any case. But we've also found out that it's not very useful to calculate just these very diverse and maybe exotic molecules if you don't have a clear rationale for that. So we need better filtering prior to those FEP calculations, and it's always very good to design such a library with a specific or several specific non-affinity optimization parameters in mind. So here's another example from a project where the chemistry is very, very challenging, and here we applied FEP in multiple rounds, looking at the R1 and R2 group. So when we started off, R1 was a substituted aromatic ring, and R2 was a small functional group, and we then performed for each of those groups larger scans on several hundreds of molecules, and indeed found different replacements. So for R1, we identified open chains as a potential alternative, and funnily enough, for R2, we found that small heterocycles could work. And the interesting thing about this is, first of all, the first part, at least for R1, that has subsequently already been confirmed experimentally that works. That's really nice, and we are very much looking forward to seeing the whole molecule. And the interesting thing about this is that when you combine all this, you will end up with a molecule that has only a Tanimoto similarity of 0.6 towards the original molecule. And one of the main criticism I've heard also throughout the years in FEP is that you only get this very similar molecules. Well, we can prove that that doesn't necessarily have to be the case. And I think when we can really navigate through chemical space like this, this is also when we can make the most impact. So let me briefly sum this up. We have learned a lot trying to use FEP in our in-house drug discovery projects. We are now certain that we have to be very careful about which systems, or very picky which systems we want to use FEP on, and we will in the future only use it in projects with excellent validation studies, with an RMSE below 1 kcal per mole and no such limitations like one R group can't predict it or something like this. We've also seen that there's still a lot of work to be done, especially we see potential for improved ligand pKa predictions that will reduce this uncertainty about the protonation state, and what would be even greater if one had FEP calculations with different protonation states in solvent and the complex. And we also saw that there's still work to be done in improving the force fields and the sampling. In terms of the operational side, we learned that in order to generate impact in projects, we see that the best way to do this is to apply FEP to large libraries when we have a specific question in mind and can push the project forward in this. However, when we did this, we also saw that we still have to do some more development in getting those, intelligently designing those libraries. And last but not least, it's good, we think it's also good to use FEP complementary to standard MedChem optimization. So rather going into front-loading, being a very active computational chemist on the project, front-loading in silico SRI exploration for series where no synthesis is currently ongoing. And also we have also started to use it for fragment optimization. So, and I will close now with our little overview of how we optimally would like to do this, starting off with either like compounds from our virtual libraries or in some other ways, large libraries, filtering them down with machine learning models or other predictive models, and then in the end having the free energy calculations and getting the best molecules for synthesis. Thank you for your attention.
Yeah, John.
This is really beautiful work, Christina. I'm very curious, in your combinatorial library organization, how much thought do you pay to what's synthetically accessible, or do you worry about that later on in the process?
That depends. So, for example, this massive library, it's designed to be synthetically accessible, but in generally, we're just starting out to consider that more and more. So there are approaches, and we're looking at software to determine this, but we're not there yet.
Yeah, Chris.
I was interested by the particular issue going from short R group to long flexible R groups. Do you know if that's because of the multiplicity of conformational states of the long R group, or is it more just going to transition from short to long?
So we get convergence, but we don't get convergence to the right answer. So I think it's really getting the right conformational state for those long R groups.
So maybe it's local convergence.
Yeah, it's local convergence. Yeah.
So as you were elaborating libraries, are you also constrained on designs or how to make sure they still preserve binding modes? How would you practically
Yeah, that's a very interesting point, and I mean some of these outliers that we are seeing in our calculations, that we have some suspicion that that might actually be the case, that it's not preserved, the binding mode is not really preserved. We cannot see that in the short time that we are typically simulating. So we are typically simulating with FPP plus only 5 nanoseconds. In some projects we extend this to 20, but usually this will not really change the binding mode throughout the course of the simulation. But yeah.
follow up? So after you run the calculation, you go back and for the ones that behave on borderline in terms of conversions and other criteria, you go back and change the two parameters on your reduced calculation?
I would love to if I had the time. No, usually not. We have to kind of use this under the deadlines we usually have. We're using it kind of out of the box, but for this analysis, then for the outliers, yeah, we try to go back and understand what's going on, but we can't do any development work.
Could you just say briefly what the FAD protocol is? How many windows are you doing? You said between 5 and 20 nanoseconds per window. How many windows?
I think it's 12 windows, and we are using just default for that. And for the charge transformation, I think it's 24 windows.
You mentioned there were issues growing into solvents with the small molecules, and those only predicted. Did you try different modern models or else different partial charge fitting methods?
No, but that would be very interesting. I mean, we now have a data set that could be used to test such models. Yeah.
Excellent. Well, let's thank our speakers again.